So our lectures are recorded and you can access them via the YouTube link right here. So all of the lectures from last semester have been purged from the folder for them, and so everything that you will now find in the folder will be actually new content. But that is our YouTube link. Special senses. Chapter 16 in the McGraw-Hill book. One of the things that I want you guys to remember, what we were talking about previously as far as sensory receptors, they were called simple sensory receptors because their construction is simple. There is a certain variation to them, but overall, when you take a look at them, they're not big. There are not a lot of things that you have to worry about putting together when you take a look at the histology and the construction of them. We are now in special senses, and these are called complex sensory receptors. So everything that we talked about before was simple, whether it was a mechanoreceptor, thermoreceptor, nociceptor, all of proprioceptor. Those were all simple sensory receptors. Now we're in the complex sensory receptors, which are called our special senses, and that's what this chapter covers with us. So let's take a look at the first of the sensory receptors and special senses that are simpler, and as we go along, things will become even more complex with these complex sensory receptors. And we're going to start out with olfaction and gustation. Olfaction is the sense of smell, and gustation is the sense of taste. We're starting off first with olfaction here, and take a look at our pictures, and then we'll go on into the notes themselves. But what we're interested in is this olfactory bulb, which is lying on the cribriform plate. You guys remember the inside of the skull and the frontal aspect of the skull? There was this area that had little holes in it, like a cribbage plate, and that's the cribriform plate. And it had a little bony triangle in the middle of it, separating the two sides. That's an important landmark for where our dura mater attaches to that. But we have olfactory bulbs that sit on the cribriform plate. That's where those guys sit. And in the human, our olfactory bulbs are really so much smaller than what you guys encountered with the sheep brain. And that's part of that is because we don't depend upon our sense of smell for survival like other animals do. So our olfactory bulb is indeed much smaller, as well as our olfactory tract. And the sheep is much larger. And what do you think about dogs? Human or sheep-like? Sheep-like. And the larger it is, that means that's real estate. That means that there's a lot more capability there. And so we have less capability with ours, but other animals with larger olfactory bulbs have larger ones. Now the interesting thing is the olfactory bulb is not where the olfactory neurons are literally found. They're literally found poking through the little holes. Those are actually the axons of the olfactory neurons. And so all those little holes in the cribriform plate represent an olfactory neuron coming up to synapse at the olfactory bulb, and therefore now we have that olfactory tract. And that's what this is actually showing you right here. It shows you this thing called an olfactory glomerulus. That's not so important, as it's important that you realize that the olfactory nerves, cranial nerve 1, or cranial nerves 1, are actually synapsing there. And so we see these guys here. Those are our little olfactory nerves right there. Now, they are emanating from a olfactory receptor cell, this guy right here, and we have axons, which we know make up nerves. We have the cell body, and then we have the dendrite. Now, what does dendrite mean to you guys at this stage? It's the receptive end. That's where things have to attach or latch on to start the entire process. So remember that. At the dendritic end of our olfactory neuron are these things called olfactory hairs. And they call them olfactory hairs because they look like hairs under the microscope. They aren't real hairs, but they look like hairs. And that's where the receptors are located. And receptors are, remember, what the chemical, because this is a chemical sensation here, what the chemical has to bind to a receptor which is located on the hairs. So that's the process. And when the chemical binds, that's called a potential, right? It's called a receptor potential. This is the same thing as little bitty numbers. Remember EPSPs, excitatory postsynaptic potentials, and receptor potentials. They're kind of the same, but EPSP is like neuron to neuron, and receptor potentials are what we refer to, what starts our action potentials in these complex sensory receptor lands, or even simple sensory receptor lands. We call them receptor potentials. Now they start the influx of ion flow into these neurons, but if you don't have enough ion flow, and you guys remember the ions that flow inside neurons is sodium in the beginning, right? To create our receptor potentials or our graded potentials or EPSPs. So if we don't have enough sodium flowing in, remember we won't get an action potential. So you have to have enough chemical causing enough sodium to flow in to where we get what's called a generator potential. Once you get a generator potential, you have an action potential. That's always in the world of our sensory receptors, okay? In the world of neuron to neuron and stuff like that, you don't get an action potential until you get threshold. So we actually associate the word generator potential to threshold. Enough of the chemical is present to actually give us a generator potential, which means we automatically get an action potential. You guys know inside your nose is a bunch of material that most people commonly call snot. And so we'll call it mucus because that is what it is when it's first generated, because it comes from mucosa. So mucus comes from mucosa, and so the inner lining of our nose is called a mucosal surface, and our olfactory epithelium, where our olfactory receptor cells are actually located, are actually up there with that cribriform plate. Well, that's up here between your eyes. And where does smell come into your cavity? It comes down in an inferior location through your nostrils. And so when you're a nose breather, which we're all born as obligate nose breathers, and obligate means that you are obliged, you cannot breathe through your mouth unless you are unable to breathe through your nose. And that's why little babies it's so bad for them to get a head cold, right? Because they have a lot of respiratory difficulty because they are obligate nose breathers. They haven't yet learned how to breathe out of their mouth. So breathing out of your mouth is a learned feature. And so when they're breathing through the nostril, it goes right over the hard palate and down out through what's called the posterior aperture. You guys know an aperture is an opening. So you have an anterior nasal aperture and you have a posterior nasal aperture. And so our air is going to flow right all the way across the inferior aspect of our nasal cavity, out the posterior nasal aperture into our nasal pharynx. We'll get into more of that anatomy later on, but that's how it actually goes in. Well, all those molecules associated with smell are not getting up here to our olfactory epithelium very easily. And so what's interesting is when you look in somebody's nose, and a lot of people don't like to do that unless there's something wrong, but when you look into somebody's nose, there's these things coming in from the side, and they're called concha or turbinates. And they increase the surface area, and they also increase the ability of the air that's coming in to kind of end up getting mixed around and vortexed. And so that helps to actually get some of these molecules up to our nasal epithelium. But there's something that we do to enhance our ability to smell things. There's a certain maneuver people will do to actually improve their ability to actually appreciate the chemical that they're trying to actually identify. And that's called sniffing. And so sniffing is short bursts of breathing in the air to create a vortex so that you actually get those molecules up here to come in contact with the receptors on those hair cells. So let's, now that we kind of talked about the whole process, let's go ahead and add some notes to this. Epithelium is in the roof of the nasal cavity. This is a poor location for smelling and or the olfaction. And so we do this sniffing. Improves possible contact. And remember, this is contact of a chemical. Cells of the olfactory epithelium are called olfactory receptor cells or olfactory neurons. We also have supporting cells, and we have these cells that are called basal cells, so they sit at the basement membrane area. And these guys are going to be called an olfactory stem cell. And stem cells are cells that can become whatever they want to be, and typically they want to be with whatever they're hanging out with. And so because we're dealing with olfactory epithelium, these basal stem cells want to become olfactory receptor cells whenever we lose an olfactory receptor cell. And our olfactory receptor cells are classified as bipolar neurons. Those hairs, again, are non-motile. And with this knowledge here that we have basal cells to replace our olfactory cells, it's important that you actually realize that olfactory cells can become damaged and therefore die. And are replaced by one of the basal cells. This is unusual because what do we know about neurons? They're amitotic. Remember, they don't undergo mitosis and they're not replaced. So we do have some areas where we actually have neurons that are actually replaced. And these chemical senses are actually an area where that replacement actually occurs. So this is unusual. It is not usual to actually replace a neuron. Supporting cells. Indeed, they actually make up the bulk of the olfactory epithelium. Their job is to produce, PD is my abbreviation for produce mucus. And this mucus is protective and helps to keep the epithelium moist. Because with the air brushing by back and forth, it's going to dry itself out. Yes?
I have two questions. So is basal cells and stem cells synonymous?
Yes. Is basal cells and stem cells synonymous? Yes, for our purposes.
And then how stem cells work? Are they collected prior to specialization? Like, is that how they work?
Well, we want to call these specifically olfactory stem cells because of their location. And so if you pluck them out and put them someplace else, because they're a stem cell, they could become something else. But you can't harvest these cells. These are not stem cells that are harvestable. These are actually part of the construction of the olfactory epithelium. As we take a look up here, here these guys are hanging out on the basement membrane. And so they are actually part of that area. And they're just sitting around waiting to actually undergo mitosis whenever they need to so that they can undergo differentiation. Because you guys know when you get a bad head cold, you lose your sense of smell. And the reason you lose your sense of smell is those nasty little virions went ahead and started shooting and killing off those olfactory neurons. And so we need to replace them, and that's what those basal cells are there for. Yes?
So the basal cells become the cell body of the new neuron?
Yeah, the basal cells differentiate into an olfactory neuron. So they'll look like these little blobs at the base, and then suddenly they'll start to grow an axon and a dendrite, and then replace the olfactory neuron that was lost. Looking at some interesting facts about our olfactory epithelium, humans can detect, it's amazing to think that we can detect 1 trillion different odors. And we can do that with only having 400 smell genes. So we can detect 4 trillion different odors, but that's based upon only 400 different smell genes. So you can imagine that this is actually a combination of smell genes. So these trillion different odors are activating different smell genes. Maybe one odor is activating only one smell gene, but maybe this other odor is activating 3 different smell genes. So we end up with a different sense of smell with that. So that's an interesting aspect, because our notes here say olfactory cells contain only one type of smell receptor, and each smell receptor is encoded for by a smell gene. So that means there's only 400 different types of smell receptors out there, because there's 400 smell genes. And so odors activate actually many different smell genes whenever they come into contact. It's not just one smell gene that will typically be activated. So smell genes encode for a receptor, protein that is, because our receptors are proteins, that are only activated by specific odors or odor combinations. In order to activate one of our olfactory cells or neurons, the chemical must be volatile. Volatile means airborne or gaseous. It must dissolve in water because the foundation of mucus is water. And remember, our olfactory hairs are actually in that mucus. So water soluble. Must bind to the hairs. So if it's not the right conformation, you will not detect it. And let me give you an example. Okay. I like asparagus. How many of you guys like asparagus? Let's see a show of hands. Some people don't like it. So I like asparagus. Okay. Well, when you eat a lot of asparagus, it makes your urine change color, right? Your urine gets kind of a greenish tinge to it. Well, the reason you have a greenish tinge to your urine is because you are actually excreting asparagine. So it is actually a chemical called asparagine. There's also chemicals called cadaverine, because that's what people associate with cadaver smells. But let's go on to asparagine. So it's called asparagine, and I think it stinks. But do you know that some of you in here do not know what I'm talking about? Because you do not have the receptor for asparagine. And so many of you are unaware of this, that not everybody can smell the same things that you smell. And if you can't smell asparagine, you don't have the gene that encodes for the asparagine odor. But if you can smell it, that means you have that. There's also this paper called PTC. Have you guys ever heard of this paper called PTC? There are tasters and non-tasters out there. And PTC, if you are a taster, and because it's just a fun test to do, is that you can give people paper that's been kind of laced with PTC, and they can taste it and they go, oh, that tastes awful. And other people go, tastes like paper to me. Well, the only reason they say that is because they don't have the gene to recognize that chemical. So the chemical can come into that area. There is no gene for it. There is no receptor. And so therefore you have no action potential or no knowledge of that aspect of your environment. So smell and taste is actually pretty interesting. Everybody thinks that we all smell and taste the same things, but that's not true, because it's going to depend upon receptor density, which means how many receptors you have for that. If you have a whole bunch of receptors, or if you have very few receptors, who's going to be more sensitive, the person with a whole bunch of receptors or the person with fewer receptors? Whole bunch. And so you're going to find people out there that are very sensitive to certain chemicals, and that means right off the bat they have more receptors in their like nasal cavity or oral cavity that are actually designed for that chemical. So it's really interesting. So our notes here say must bind to the hairs because that's where the receptors are located. And then receptor potential. So if I put one molecule there and it opens up the sodium gate for at least 2 sodium, ions to flow in, that's a receptor potential. But if we are not aware of it, we have not yet created that generator potential. Because once we get enough sodium ions flowing in, we get a generator potential and then always an action potential. It's always in that sequence. So the first thing to always happen is a receptor potential, and if it's strong enough or enough chemicals, then we get a generator potential and an action potential. Taste buds, the next chemical complex sensory receptor. So we associate our tongue as the area where taste buds are located. And that's true for the most part, but we're also going to learn that it's actually also, we have taste buds in our pharynx. And our pharynx is the back of our throat. The term papilla. Papilla is a projection. Remember, process in the bone world was a projection or a protuberance. So now we have another kind of a histology term called papilla. So papilla is a projection, and whenever I think of papilla, I just typically draw it like this. Each one of these upward guys is a papilla, To me, that's a papilla. But they're not always exactly like that, because remember, it's a protuberance here on a microscopic level. And there is a little bit of gross anatomy level, too. So our notes are talking about papilla and taste buds because that's where taste buds are located. They are located with certain papilla of the tongue. The tongue has 4 papilla, and the 4 papilla are filiform, fungiform, foliate, and vallate, also referred to as circumvallate. So there's 4 different types of papilla or projections on our tongue. The most common papilla on our tongue is actually the filiform papilla. That's what gives our tongue the relatively what I would like to equate to a velvety appearance. It's a roughened or velvety appearance to your tongue. That's from the filiform papilla. They have no taste buds, and they actually make up 2/3 of our tongue as far as papilla goes. So the filiform papilla have no taste buds. And in the oral cavity, what is the lining of our oral cavity? What do we call that? What do we call the lining on our skin? Epithelium. And let's give it a little bit, be more specific. Stratified epithelium. And it's keratinized on the outside. What about on the inside? It's non. And on the inside, it's stratified, and it's not simple because on the inside, that's also where we have high abrasion. So the keratin covers the papilla. You guys follow me with this? Not keratin. Skin that is non-keratinized is covering the papilla. So stratified squamous epithelium is covering all of these papilla. And our taste buds are located in the epithelium on the papilla. So the papilla is a protuberance. They're all covered with stratified squamous epithelium, and to find your taste bud, you have to look in the epithelium to find them. Now, the 3 papilla that do have them, the fungiform, foliate, and vallate papilla, the taste buds are located in specific areas of the papilla. And so you don't have to worry about finding them with the filiform, but if you want to find them with the fungiform, the taste buds are at the top of the mushroom. Fungiform, mushroom-shaped, is what they thought when they looked at it. And if you're a foliate papilla, you have to go in around the inside of the projection. So go to the base of the projection. And so you have to look for them in these areas. And then if you're the circumvallate or the vallate papilla, this is where you actually find your taste buds. You guys have, for a histology slide, that's what you guys have for histology is the vallate or circumvallate papilla to locate the taste buds. So you can't find them if you aren't looking in what I call the crevice area. If you go to the top up here, You know, you'd have to, in order to find that taste bud, it'd have to be a fungiform papilla. You won't find it at the top. You always find it in the base. So you have to go in with the microscope and you have to look in these areas in order to actually find them from the top on down. So we have some cartoon pictures of histology of what a taste bud looks like. And the taste cell or neuron is called a gustatory cell. Gustation is taste. And they also have a hair that comes out. Here it's calling it microvillus. We also call it a gustatory hair. So let's keep with our other chemical sensation, that special sense that we talked about, which is olfaction. And that comes out of this thing called a taste pore. So as we're taking a look at this, you guys can see the stratified squamous cells on either side of this taste bud, correct? And the hair is coming out of the taste pore, and just like with olfaction, that's where the receptors are actually located. And so our chemical has to come into contact with the receptors on the hair in order to actually activate the gustatory cell neuron and have the sensation of taste being conveyed back to your brain. This is a histology view of that cartoon, and hopefully you'll be able to make the transition from the histology cartoon as well as an actual histology slide of that. So the pores here, and you can't see the hair. If anybody ever sees a hair, I want to see it because it's just so small. It's not something that you would actually ordinarily see on a preserved specimen. So let's now go ahead from the picture. Let's go to the notes. Papilla, visible bumps on the tongue. So that means you don't need a microscope to see them. But what about the taste buds? Do you need a microscope to see the taste buds? Absolutely. You need the microscope to see the taste buds because you have to look into the epithelial layer. So types of papilla or projections: filiform, short spiked, anterior 2/3 of the tongue, and their function, so no taste buds on them, their function is to actually manipulate food. So when you take a look at the picture up above, kind of look at them as like little teeny tiny razors or a serrated knife. It's designed to actually kind of help break things up so that we can break them up so that they can then ultimately get in contact with that hair so that we can actually appreciate that taste. The fungiform papilla is mushroom shaped, and so what we will do with the fungiform, we will find the buds on the top of this papilla, not at the base. And as far as fungiform papilla, you will actually find them more concentrated on your tongue. They're not an obvious, they're not a whole lot of these guys, but they're more than the folate and the circumvallate. They are concentrated at the tips of your tongue and sides. Your folate are really strange. They are found way back in the oral cavity, right in this area. So close to the base or the back of the tongue, lateral to our vallate or our circumvallate papilla. And so there's the vallate papilla are very unusual. They're very large, very easily seen, and that's where we find those taste buds on the foliate and circumvallate in the sides of that. You guys know what these guys are, right? Off to the side? Tonsils. And so that's lymphoid tissue, and also at the back of the tongue is a bunch of tonsillar material. It's lymphoid material also there. That's what that picture is all about. Adenoids sit on top of our soft palate. So those are the ones that cause people to snort when they get them swollen and enlarged. But folate, remember the buds are in base. Circumvallate, buds in base also. And these are the largest of our papillae, and they form an inverted V. at back of tongue. There's a typo on my part. Sorry about this. There are 3 cell types here in a taste bud, just like there were 3 for the olfactory epithelium. We have gustatory cells, which are our chemoreceptor cells, and form the bulk of the taste bud. Then we have supporting cells, and they support and sustain the gustatory cells, help to make sure they stay healthy. And then we also have the same process here, basal cells acting as stem cells to replace our gustatory cells whenever they die. And remember, we have a gustatory hair. Projects from pore and has receptors for various chemicals. What you may not be aware is that we actually have replacement of our gustatory cells every 7 to 9 days. And the older we get, the replacement time, what gets shorter or longer? Longer. So we'll stop there, and I'll pick up the pace, and we'll get through most of vision next time. Wow.